Introduce both of our speakers. Welcome to the webinar series. I'm Professor Michael Strano, lead PI for the Disruptive and Sustainable Technologies for Agricultural Precision (IRG), or DiSTAP. It's my pleasure to introduce our two speakers who are going to have a tag team presentation. Dr. Gajendra Pratap Singh is our scientific director at DiSTAP. He is a principal investigator as well. After his PhD in Spain, he worked at MIT developing portable and wearable Raman spectrometers for Bayer Healthcare, Samsung, and DARPA. He co-founded and led a few startup companies in the UK and Spain before moving to Singapore. And then Dr. Minho Wang is actually our deputy scientific director at DiSTAP. He's been a part of the IRG since its inception. He's a research affiliate in the Department of Chemical Engineering at MIT. He was named a Forbes 30 under 30 and MIT's Tech Review Innovators under 35 for his work on the development of nanosensors for use in plants. He was formerly a project leader at Boston Consulting Group, serving clients in the agri-food and specialty chemical sector. And so the two of them are going to lead a presentation on tools for next generation agriculture. So with that, I'll turn it over to our speakers.
Yeah, thank you, Michael, for a kind introduction. I welcome all the guests and would also like to thank the organizers at the National Research Foundation and the Singapore MIT Alliance SMART. It is a pleasure to be part of the CREATE webinar series. For those of you who are new, Center for Research Excellence in Technology Enterprise, CREATE is a center created by the National Research Foundation, Government of Singapore, where the best institutions, the global best institutions are located, and they collaborate with institutions. For example, we have MIT, Singapore Alliance, we have Yale Singapore Alliance, Berkeley Singapore, even Shanghai, Jiaotang University, and Singapore Alliance. We all have our own ecosystem and we all develop our own footprints, but we also collaborate with each other. So in today's talk, Tools for Next Generation Agriculture, I will speak about the analytical technologies that our center is developing. And these analytical technologies are one of those technologies that will be needed to increase agriculture production. Basically, if we want the food supply to keep in par with the population growth and climate change, then these analytical technologies are one of those technologies that will be very useful. Let me start by saying a few words about what DiSTAP is. Our center, DiSTAP, is a collaboration between Temasek Life Science Institute of Singapore and the Singapore MIT Alliance. In Temasek, in the TLL, Temasek Life Science Research Center, we have the plant biologists, and in the SMART, we have the engineers and scientists, and we both work together in a multidisciplinary team. And this is our team, the leadership team. So Professor Michael Strano and Professor Chua are the lead PIs, and we have Dr. Zhang working in shade avoidance syndrome, Dr. Zhang Chao Yin working in heavy metal detection, Dr. Sorojan working with volatile organic compounds, Dr. Daisuke in plant breeding, Dr. Park working with nutrient detection, and Professor Strano develops the nanosensor technology, Professor Ram develops the Raman technology, Professor Marelli develops the biomaterial technology, and I work with all of them to run the center. Now, besides these technologies which I talked about, nanosensor technology and Raman technology, which are basically applied to plants, we have other NRF-funded projects as well. For example, with the A-STAR and the TMSI, we have detection of heavy metals in the fish farm. With the Duke NUS, we have development of tissue biopsy device. So with the Duke NUS and Singapore General Health Hospital, what we are doing is we are using the DiSTAP technologies to detect liver cancer, for example. And then with Central Narcotics Bureau and Department of Biological Sciences at NUS, we use the Raman technology to detect drugs in real time. So So basically, these analytical technologies, which I will talk about today, they can not only be used in plants, but they can also be used for other applications. So I will look forward that some of you will get back to me for your particular applications. Now, we talk about climate change, we talk about population explosion, but really we should actually focus on the 2050 food crisis. And the reason is that as India and China, which are one of the most populous countries of the world, as they grow in population, and because of climate change, the resources decrease, there will be shortage, there is a predicted shortage of food. So until now, because of the Green Revolution, India basically made more food than was required and actually exported food. But it is predicted that by 2050, things will change. So as you can see here, that the total cereal produced has to keep up with the world population. And the food prices are also rising. This will also create a problem because people would like to buy food at reasonable prices to have affordable food. So how do we supply extra food for this 9 billion population 2050? So one of the aspects is we can reduce food waste. Food waste is a big problem. And besides that, if you look at the reasons for crop loss, like diseases, insects, and weeds, we should also be working on removing these aspects of the reduction in the production of crop. Then we know that water accounts for a large volume of large volume of material which is required for agriculture. So like 70% of the global water withdrawal is for agriculture. And we see on the graph on the left that some of the materials, some of the items which we can avoid to save water. So basically, if you avoid eating chocolate, maybe you're helping to avert the 2050 food crisis. Then came the first agriculture revolution, where it happened in Britain. And what happened is, because of the machineries, the production of food suddenly increased. And this then came the second agricultural revolution, because population kept increasing and we needed more food to sustain ourselves. In the second agricultural revolution, which is known as the Green Revolution, we used more fertilizers, more herbicides, more pesticides, and high-yield seeds like dwarf wheat. Norman Borlaug introduced it to India, and India became self-sufficient because of that, as you can see on the graph on the right. And because of this Green Revolution, many countries became self-sufficient. And we could avoid, again, the shortage of food. But as we predict what is going to happen next, what we are seeing is, again, the production is not keeping up with the population increase and the climate change. So let us say, because of some scenario, because of rising food prices or climate change, there is a lack of food. Then what should we do? So these are the challenges which we have to encounter. So excessive use of fertilizers during the Green Revolution, excessive use of pesticides and insecticides, they have made land barren. Because of climate change, the water availability is less, the population is rising. And a recent development which has opened our eyes to pandemics. So in pandemics, the borders are closed. In one area, the food is being wasted while it cannot reach to the other area of the world. We need sensors. So in a precision agriculture or urban farming scenario, you require a decision support system. And this decision support system requires lots of sensors. Now, when you look at those sensors, you can have sensors for temperature, humidity, light, etc., which only measure the environment. They don't measure the plant itself. So for example, if a human is sick and he goes to the doctor looks at the person and he tries to treat the person itself rather than measuring only the surrounding. So we have to have the same approach. And this is where DiSTAP sensors take effect. So on the left, you have a graph which shows that in a plant, different stresses generate different kinds of responses. So for example, plants are an intelligent system. For example, if there's a bug bite or there is high heat, the signaling is different in both cases. If you can detect that signaling in the plant, then you can predict before what is going to happen and basically stop your crop getting lost. So if you look at the farmer, what the farmer does is he looks at the color of the leaves, for example. But sometimes it's too late to see at the color of the leaf, and it cannot be reverted back. So if you use intelligent sensors, analytical sensors, then you can take action in time and save the productivity of crops. Now, if you look at the state of the art, how the plant biologists currently measure the plant biochemicals. So our DiSTAP analytical sensors measure the plant biochemicals in real time, because they need to measure what is going to happen inside the plant, in the plant growth or infection or the stress in the plants. So if you look at the current state of the art, the plant biologists use very bulky machines, which requires a lot of expertise, and it's very costly. And the results don't come in real time. For example, you may have seen they take plant tissues, they grind it down liquid nitrogen, and they run it through HPLC or LCMS, GCMS machines. What we do at DiSTAP is we use real-time nanosensor and Raman technology, where in the case of Raman technology, a laser interacts with the sample and the scattered light acts like a fingerprint. While in the nanosensor technology, a nanosensor is coated with a polymer, which is specific to a certain hormone in the plant, and it interacts with the plant, and you can find out what kind of hormone the plant is producing. So on the left top, I show you the result from one of our new novel Raman sensor developed at DiSTAP, which can look at which can look at secondary metabolites with more sensitivity. On the left lower section, you can see how nanosensors can also be used for gene delivery. So basically, CRISPR-Cas9 system can be complemented with nanosensor technology. On the right lower section, you can see that a plant leaf, when infused with, when infiltrated with nanosensors, which are coated to interact with a certain hormone, they give a fluorescent signal, while the other part does not. So on having doing an image of this leaf, we can find out what kind of hormone the leaf is making. On the right upper part, we have the biomaterial technology, where silk has been used to make novel material. So I will talk later that how silk-based material can be used to preserve the food for a longer time. Now, my colleague, Dr. Min Hao, will explain you about the nanosensors. Now, Min?
Yeah, so let me deep dive into a few of the technology areas. I think the first one that I wanted to share a bit more is the nanosensor aspect. So the nanosensor piece was really a multi-step development. At its very core, it's something we call corona phase molecular recognition, or COFMOR. And this technology is really something that was developed at the Strano lab at MIT. And how this works is we first start developing what we call amphiphilic polymers, which are polymers with hydrophobic and hydrophilic ends. These polymers, when they interact with the nanotube, will wrap around the nanotube to form something called the corona, which is the picture you see in the middle. And if you look across in the middle of the page on number 2, when you have this corona phase that is wrapped around the nanotube, when analytes of interest, such as plant hormones or other interesting analytes interact with this corona phase, it will cause changes in the dielectric environment. And through that, the fluorescence signatures will change. So that's number 3 at the bottom of the page. When there's this interaction, the wavelength and intensity could change. So the intensity could drop, or the wavelengths could shift depending on the interaction with the analyte, between the analyte and the corona phase. So this is the core technology that is being used for the development of nanosensors. And what we have done is to infiltrate this nanosensors into the leaves and tissues of plants to enable us to detect things in a real-time and non-disruptive manner. Next slide. So after this core technology was developed, we worked closely with colleagues at TLL and NTU and so on to start putting these in the leaves and tissues of the plant to see what information can be obtained dynamically from it. So what you're seeing on the left is a series of false colored images showing the quenching response of carbon nanotubes that have been functionalized through carefully designed polymers. So this picture labeled A here would be a leaf. You can see the silhouette of the leaf, infiltrated with two kinds of sensors. One, what we call the G SWINT, which is G single-walled nanotubes, is the active one. So there's a red color arrow. And the blue colored one is an invariant reference sensor, which will not change in the presence of analytes. So the whole purpose of this is to have one stable reference sensor and one that will respond to your analyte of interest. So over time, as you can see, time equals 0 minutes all the way until time equals 20 minutes. The red colored one, which is the active sensor, quenches and then bounces back, while the blue one, which is invariant, remains constant. So that's the figure on the right side, where the G carbon nanotube, which is the active sensor, detects the analyte of interest and then recovers, while the A single-walled nanotube, A SWINT, is the invariant one reference sensor. So over time, through using this ratiometric approach, where you have a reference and an active sensor, we can actually detect a time profile of sensor response for wounding of the leaf. So H2O2, the analyte which the red colored sensor is responsive to, actually tells you whether the leaf is wounded or not. So this is what we find to be a rather elegant example of how these sensors can be used to give you dynamic responses. Next slide, please. So basically, after we prove this, is workable in the lab scale, the next thing for us clearly is to make sure that we can enable this in the field. And here is an example. I think the pictures probably tell a story. We infiltrate the sensors into the leaf of the plant. We design a low-cost Raspberry Pi module, which I think a lot of people here will be familiar with. It's a cheap $20 camera that we adapt the camera system for in the near infrared range. And then we use it to detect at a distance the two carbon nanotubes that you've seen in the previous page. So this would allow you to actually read off signals from the plant at a distance. So you can imagine, querying your plant with your smartphone, for example. And through doing that, you can actually start imagining a situation where you employ these sensors out in the field or in the urban farm setting, where these plants can actually tell you in real time how they're doing in terms of, are they being infected by pests, or are they actually growing very well. So I think there's a little video on the right side which Katrina could play that just shows. Oops. I think that just shows an example of how in real time the invariant and live signal responses. Yeah, I think if it's not playing, I think it's fine. We can move on.
Yeah, Hyman, thank you. The video, yeah, so the video shows the nanosensor images.
Yeah.
So this is an example where we have used Raman sensing to detect plant stress. So on the left to the top, we have two leaves. One plant was kept under nitrate deficiency, and the other one was kept under proper nitrate conditions. So we take the leaf and we measure the Raman scattering from the leaf using our system. Similarly, in the middle, we have full light and shade. So when the plants are kept in different shade or different light conditions, then they grow differently. They show some stress if they do not get enough light. And you can detect that stress in real time using our Raman sensor. On the left bottom, you can see a plant which is healthy and another one which has been infected with a bacteria. And before you can see by eyes if the plant leaf is infected with bacteria or not, or before you can see by eyes, or a farmer can predict if this plant is infected or not, our Raman sensors can predict within a couple of days if this plant is going to get sick. And now on the right, I show you the Raman spectra from all these plant samples, and they act like a fingerprint. and you can detect in real time. The next part of our work is detecting essential oil in a plant trichome. For example, mint and basil, they have trichomes in them, and these trichomes have essential oils. So we can, in real time, detect the quality of oil in these trichomes. This can be extended to marijuana plants or other kind of plants where trichomes have medicinal value. So I can show you on the right bottom. I'm showing you a Raman spectrum acquired from the top of the trichome and from the side of the trichome using our systems. Now, all this spectral data and imaging data we have from nanosensors and Raman, and it can all be combined and sort of using Internet of Things or Internet of Plants. You can use AI and predict in real time what kind of stress a plant is going through. So this is an example where AI was used only on the images of the leaves, and it has been shown that just by studying these images and using machine learning and artificial intelligence, you can predict the diseases in the leaves. So similarly, what we are working on is using our spectral data, making databases, and comparing with those databases and trying to predict if a plant is under stress. We also work with biomaterial technology, where silk-based biomaterial is used to preserve the food. So on the left, I show you a strawberry. If this strawberry is dipped in the solution of silk, then it remains fresh for more time than if it is not dipped. It's kept as such in the shelf. On the right is an example of avocado, where if you keep an avocado normally outside, it gets oxidized, while if you dip it in a solution of silk biomaterial, it remains fresh. It looks fresh for a longer time. This will be extended to include nanosensor and Raman technologies, where micro needles made of these silk materials will be used to inject carbon nanosensors or other materials into the plant. And this is how we can get real-time data, real-time plant hormone data, and secondary metabolite data from the plants and predict the plant stress. This is an example I'm showing you how we miniaturized the system, the Raman and nanosensor systems, and made them portable. And we work with the local industry, TechnoSpecs, and we help them build new versions of the instruments. So this is one version of the instrument made in Singapore, where our TL colleagues are using this instrument to detect plant stress in real time. We built this clip, which you are seeing with a 3D printer here in DiSTAP. Now, Min will take you to the market analysis of our technology. Min?
Yeah, so thanks, Kajendra. So basically, this slide is clearly, a lot of logos and a lot of things happening. But I think what it tells you really is that the agri-tech market is really very vibrant. There are many startups, popping up in different aspects of the value chain. I think what DiSTAP is trying to do is to focus on areas that we feel our technology can make a difference. And specifically, it's in the area of plant data analytics, the development of sensors, and how do we use it to predict things in the case of precision agriculture. So next slide, please. So just if we take a step back, all the technologies that Kajendra has shared, independently, I think if we all put them together, what we are really trying to do is to move from the model plant. So that's the left side. In the lab setting, very often people work with plants like Arabidopsis or tobacco, which are well understood, or the genetic composition is very well understood already, and really try to use some of our tools to bridge the gap between the model plants and the agriculturally relevant plants. So this figure, we published it in Nature Plants, but what we're trying to do really is to use math models, data science, develop Raman nanosensors and other tools to help move from the model plant situation to really applying that in the field or in urban farms, where we can start applying it to your bok choy or other crops that Singaporeans eat and which are agriculturally relevant. Next slide. So more specifically, some of our technologies, we have started to think of form factors that can be applied to the urban farm. So on the left side, you see some of the form factors that we have started to think of, such as the nanosensor optode, which we got an innovation grant award for, or trying to miniaturize some of the handheld Raman systems that Kajendra has shared, and start thinking how can we apply this to the urban farm. So working very closely with TLL and developing these form factors to make sure the use is simple and accessible to farmers who come from a different background, and not all, they always prefer a much simpler form factor. So if we manage to get these sensors in a form that can be applied easily, this would potentially change and improve the cycle where you can grow crops, intervene using these data quickly, adjust the growth conditions in a resource efficient way, and accelerate the yield and time to market. Next slide, please. So more specifically, what we have done also is to start comparing our technology to what's out there. Basically, what's the gold standard, and how does DiSTAP technology compare? Where are we stronger and where are we weaker? So I won't go into a lot of the details, but I think there's some factors which farmers always ask about, and that is, if the data is static, meaning what we collect is only useful at a certain point in time, or can it be dynamic, which is real life information? How fast is the collection? So that's the speed. How sensitive for the particular analyte of interest? Is it non-destructive, meaning do we have to cut up the leaf to measure it, or can we measure it without damaging the plant? How easy to use it, and of course the costs. So these are all things that we are very cognizant about as we try to develop technologies for use in the urban farm.
To Dr. Singh and Dr. Min. So the analytical tools, inclusive of these nanosensors and the fiber-based leaf interfaces, right? The leaf clip interface. So this has been applied to certain types of crops, such as, if I can gather correctly from the scientific reports paper, it's rice and tomato. So has this been applied to other field crops, such as corn or wheat, or even to those crops grown via controlled environment agriculture, such as currently being carried out by Sustainare in Singapore? Because I recollect Sustainare because I met with the CEO and the co-founder of this company, Mr. Ben Swan, at the sustainability conference at Marina Bay Sands. So I was just wondering if this technology has been tested or applied to crops grown in a controlled environment, which is very relevant to markets such as Singapore.
Yeah, hello, Nom. Yes, so we are now planning to expand into the area of measuring plants which are grown in the field, like corn. Okay. We also are planning to apply the technology to rice, okay, besides the plants which are grown in an urban farm setting.
Oh, okay. And my last question, quick one. So to implement this impressive piece of interdisciplinary technology, as I would put it, which includes 3D printing and spectroscopy in agriculture, that has resulted in a prototype gadget, which is, a compact Raman sensor, I would say. So this requires extensive reliability and technological robustness, but from my experience in tech launch at NUS, moreover, above these two, it requires economic or financial feasibility for scalability. So what's the current status or your opinion on the financial feasibility for this technology for a large-scale implementation?
Yeah, Min, you take it.
Yeah, so I think you could see, we're starting to think in that direction where I showed an example of portable Raman system.
Oh, yes, yes.
And of course, yeah, of course, if you produce these at scale, then our assumption of $12,000 a unit could actually be lower. Okay. We have also made some assumptions on how much time it takes for each data point or each loan to be taken. There's some variation. I can go into the second order details, but basically there's some variation depending on the type of crops and how the farm is structured and so on. But at a high level, I think we are quite confident that there's still a sizable economic margin for the technology to be used in the field for your average edible crop, at least within the context of the developed countries.
Okay, so that's it. Dr. Singh, Dr. Min, thank you for the talk. It was really very informative and very impressive. I've also come across the Nature Plants paper, and it's an excellent article. So hope to meet you soon sometime around. Thank you so much.
Thank you, Anup. We have several questions in the chat window, and I thought I would help facilitate by reading some of them aloud. So Tang Wei Lin is asking, curious, does the nanosensor remain in the plant and get consumed? Any harm to...
Yeah, we can't hear you, Tony.
Oh, hello, can you hear me now?
Yeah, so
Yes.
I think Min replied to that. So yeah, let me explain. So although nanosensors cannot be eaten, let me be clear, but there is way to contain them. It is not necessary that you infuse the plants with nanosensors. So in the development of the technology, where plant biologists want to know what biochemical reactions are going on in the plant in real time, you need to infiltrate the leaves. But in an urban farm setting, these nanosensors can be contained in a microneedle, like let's say silk microneedle, or in a gel-based matrix, and then which can act like a sticker or a strip, and you touch the plant leaf or touch a fruit and get away with it. So no nanosensors seep out into the fruit or in the vegetable, but then you can use that strip to have the nanosensor image. So yes, you cannot eat the nanosensor, but of course you can use those nanosensors in a gel or in a contained matrix to still detect plant hormones or pesticides or insecticides. Even in aquaculture, you can use it. You can use in cancer research. So there are lots of ways you can use this nanosensor technology.
Thank you. Cheryl Tan was asking if the technology has been trialed at our local urban farms in Singapore. If so, what has been the feedback so far? I saw some of the local companies mentioned on a slide a couple slides back, but maybe if you could expand upon that for Cheryl.
So until now we have.
Yes, sure.
Yeah, Min, take it. Sorry. Yeah.
Sorry, go ahead, Gajendra.
Yeah, so Min, I was going to say that we have used it at TLL in an urban farm setting. You have more data?
Yeah, so I was going to say, yeah, exactly. Like we have mainly trialed it with work closely with our TLL colleagues to test some of these systems. What we are doing with the actual, commercial urban farms is we have engaged in talks with them to potentially start working with them to also start piloting some of these out in the commercial setting. That's kind of where we are at right now.
Okay, thank you. And that was from Cheryl Tay from the Straits Times, I believe. Good. The next question is by Daniel Wong. Why can't we implant more agricultural plants to buildings in Singapore when we are planting so much landscape?
Tough question. Not sure if Gajendra and myself are best positioned to.
No, I will be. No, I will be happy if they do. I will also be happy if they allow in some common area cows and other domesticated animals, so that we cannot throw food but give to them, right? Sustainability. But yes, so we can implant more agricultural plants, but I think here decision has to be made from the government, and I think we can put this idea out. It's a good idea.
Yeah, I think there's always trade-offs, right? Some people use the rooftop for solar. Some people advocate for urban farm rooftop farming equivalent. So I think it could be a case-by-case thing, but it's definitely something worth taking a look to examine the trade-offs. Yeah.
Yeah, and there's some interesting local farms popping up, or at least community farming, even next to some of the HDB blocks, like in Haji Lane. You have plots where community gathers and grow plants. Next question is actually there are two questions pertaining to the silk dip to preserve fruits and vegetables. So the first is by Pooja Agarwal. Impressive talk, thanks. Does silk dip change the taste of strawberry or other fruits to which it's applied? And the second question is by Yu Chen Long. Would applying silk coating, for example, by spraying to growing crops, protect them from stressful conditions?
Yeah, I think on the taste part, we are working on it. It so depends on the taste buds of the person who's testing them. But of course, it does affect the taste, and but it is not toxic. So basically, it's a natural material, so it can be eaten. Now, a natural strawberry, completely natural or coated with silk, there may be some differences, but I think it can be worked on. It can be changed. Now, can you spray silk material on a plant? This is something I can get back to you by email. You can write to me because I'm not sure if first of all it is feasible to do it, because how much part of the plant will you cover by spraying? Maybe some parts will be not covered. So I can get back to you after talking with the experts in the field.
Okay, and the next question, again by Pooja Agarwal. Also, how early Raman technology can detect pest infection in field plants? In other words, at what stage of the infection can Raman technology detect the presence of the infection?
Yeah, so in our current work, we can detect in 48 hours or earlier than 48 hours. We have a signal in Raman where we can predict that, yes, this is a bacterial infection or a fungal infection. Yeah.
Okay. We've efficiently moved through all the questions in the chat window. So again, we can open the floor to any verbal questions as well. I see one additional question. Oh, go ahead.
Hi, Tonio. Kiang Wee here. Hi, thanks to Minhao and Gajendra for nice presentation. You know, in the last few days, we read about a whole new slew of projects on food technology, food science being launched in various parts of Singapore. Is the technology you describe, is it feasible to think of offering the technology you describe as some sort of enabling tech to many of those initiatives to facilitate them in their research? Thank you.
Thank you. Yeah. So, sorry, I didn't get the question. So, yeah, it is enable technology. So, can you please say it again?
Sorry. Yeah. You described a very interesting technology that allows you to sort of sense various things earlier than you otherwise could. At the same time, we note that quite a number of new projects in the on urban agriculture that were announced in the last week or so. I was wondering if you would be interested or could explore how your technology might help those projects get going with get going faster and perhaps with more capability than them developing independently might. Thank you.
Yeah, sure. Yeah, thank you. Yeah, we'll be happy to explore the possibilities of expanding the domain of the application of our technologies. Thank you. Yeah.
And actually, if I can just add a bit, I think both Gajendra and myself, as well as the broader team, TLL included, we do interact with some of the other players in the ecosystem quite a bit. So we do really appreciate working closely with any other players in the ecosystem as well. So if anybody on this call, have names or have particular partners that you think can draw the link, there'll be super appreciated as well, for sure.
That's very good. Thank you.
So I saw one question in the chat window that I had earlier overlooked. It's by Itai Herman. Thank you for the great presentation. How is the Raman data collected? Is it in contact with the leaf or away from it? And perhaps an extension of this, if you could speak more generally to contact versus contactless methods of data acquisition. For example, how much of this could you mount on a drone and fly over a crop as opposed to getting in direct contact with the plant? And what do you sacrifice in terms of accuracy of the information and sensitivity?
So right now, most of the data I have shown is collected in a contact basis. But right now we have a system which can detect from a distance of 10 centimeters. So we have the capability of having both remote sensing as well as contact touch Raman sensors. Now, when it comes to drone, there are other factors. We need to miniaturize the system more. And also, because it's a laser, it's not a light source. So we need to understand how much laser power will be used. And again, the regulatory affairs come into play. So drone is still a bit far. We haven't applied it on, we haven't used it on drones yet. The Raman sensors we haven't used. But the nanosensor imaging part, I can see it can be used on drones. Because the fluorescence is almost 1 million times more stronger, I will say, compared to some weak Raman signals. And you can detect the fluorescence from these nanosensors from meters distance. So I see that, yes, thank you for this idea. We are working, actually we did discuss, brainstorm about these possibilities. Yeah, thank you.
Thanks. We have a few more questions popping up in the chat window. One is by Mien. Understand that each type of plant needs a specific sensor. May I know how many types of sensors have been developed? How fast in developing a sensor for a particular plant?
Yes, so
maybe I take that question.
Nanosensors. So Min, can you please speak on nanosensors?
Yeah, sure. Yeah, so actually for the nanosensors, I think one of the beauty of the technology is that it's actually not specific to the plant, but it's specific to the analyte. Meaning to say, if a plant has, like a particular hormone, auxins or jasmonic acids or whatever, it's actually specific to that analyte, that hormone. So as long as the plants that you're applying this to you're able to find a way to infiltrate these nanosensors within, you can actually apply that, broadly. So this is, I think, one of the advantages of the nanosensors compared to some of the more genetic approaches that are out there. As to how fast developing a sensor to a particular plant, I think at the initial phase, to be honest, it took us a long time because we had to develop the polymers to in the right buffer system and try to apply in the plants and then try to find a way to read it off at a distance. I think now we have a better system and working closely with our partners in Singapore as well. So I think the main part that takes us a bit more time is still in the polymer discovery phase. But once we get the polymer construct discovered, then it's actually the rest of the steps, could be potentially quite fast. What about the Raman side? You want to add anything?
Yeah, so one of the advantage of Raman is that in one single shot, it can detect multiple analytes. Of course, you need to choose the laser, whether you want in the visible or near infrared or ultraviolet. So you have a series of wavelengths where you can choose the laser from, but the Raman signal never changes. From a certain molecule, it's so highly specific that if you have glucose and fructose in front of you, both white powders, it can tell you in a second that this is glucose and this is fructose. So a single shot of Raman spectrum contains information about multiple analytes. There can be tens of analytes in there. And you can use machine learning to extract them. Sometimes you can extract them by univariate analysis, depending on what kind of signal is coming out from the Raman data.
Thank you. And just to combine the two last questions into one, we'll make it our final. The Raman sensor, what are the potential applications in A, food quality measurements in supermarkets, and B, outdoor crop in the field?
Yeah, food quality is a very interesting subject where we would like to apply these Raman sensors. So you see, in an ideal scenario, if you are having wine, and you can use your Raman sensor built in your smartphone, and you can find out if it is from Chile or from Spain, then that's an application which you'd like to have built in your smartphone. And there are Raman sensors now which are integrated with a smartphone, but the database part of it, the Raman spectral database part of it, is not that well developed. So yes, in food security, in food safety, this technology is it has very high potential. And when you say food market or where, I think instead of that, it should be applied as a technology for the supply chain safety. So, for example, if you have food coming in the food market, then the manager can basically use it just to randomly check the authenticity of the food.
Wonderful. Thank you. And thank you both. I look forward to integrating your sensor into the latest smartphone so I can go to the supermarket or to a restaurant to know if I'm getting mackerel or soulfish. Finally disambiguate that in my mind. Thank you for the great presentation today, the great seminar. Also, Kyung-Wee Lim sends his regards, saying a great seminar. Thank you. And thanks all for attending today and for taking time out of your day to learn about the great work going on at DiSTAP. This officially concludes, but if the speakers want to hang around for a few extra minutes to handle Q&A, that's fine too. Otherwise, I wish everybody a good day, or Michael Strano, a good evening. Thank you for coming from MIT, and we'll see you all soon.
Thank you, everyone.
Thank you. Please write your questions in an email. You're welcome.